Hello everyone. So today we are going to start with first we will do this mock test and then we will practice some practice question from the chapter Biotechnology and its application. So let's see the first question. What the first question says: which of the following best describe the term totipotency in plant tissue culture? So they are asking us what the term totipotency, why this term totipotency is used in plant tissue culture and what its importance is, what it describes. So, options are ability to form a whole plant from any cell or explant. Second option says formation of somatic hybrids through protoplasm fusion. Third, production of virus free plants using meristem culture. And fourth, development of pest resistance crop via genetic engineering. So they have given us four options and let's see each and every option. So the first option says the totipotency in plant tissue culture means ability to form a whole plant from any plant or cell. So answer is option 1 only, but we will discuss it more. So totipotency itself, giving it it's the name itself is giving us the idea that toti means total, potency means has a potential, means any part or any cell of the plant has the potential, has the ability to form a full whole new plant, a total plant, a new plant. That what list totipotency describes. If you guys look at the other options, so they are like formation of somatic hybrids through protoplasm fusion. No, this is not what totipotency implies. This is used in somatic hybridization and all. Third, production of the virus-free plant using meristem culture. No, it does. It doesn't also means like not describing totipotency. And development of pest resistance crops via genetic engineering. This is also not describing the totipotency. So the answer is first, ability to form a whole plant from any cell or explant. Next question says, how does PCR aid in early diagnosis of infectious disease? Means how does PCR, what is PCR? Polymerase chain reaction. So this how this polymerase chain reaction is responsible, how we can diagnose any infection, any infectious disease very early using polymerase chain reaction. So options are first, it increases the concentration of antibodies. They are saying that we using PCR for early diagnosis because what it does, it increase the concentration of antibodies. No, it doesn't do that. Second, it amplifies the pathogen's nucleic acid for detection. So we all know that what is actually PCR does. So PCR will amplify the number of nucleic acid, that could be DNA and RNA. So here what it can do, it can amplifies the pathogens, that infection causing organisms, pathogens. They can amplify its nucleic acid, that is RNA or DNA, for detection. So the answer is option 2. Let's see the other options. It identifies mutation through autoradiography. No, it doesn't do that. Fourth, it measures antigen and antibody interaction. No, this is something ELISA do, that it is depends on antigen and antibody interaction. PCR only amplifies the nucleic acid. So answer is option number 2. Third question says, in ELISA, which is enzyme linked immunosorbent assay, detection of a pathogen is based on, this is something I have told you in previous question also. So this, how we can detect pathogen, on which principle this ELISA works. Options are amplification of pathogen's DNA. No, this is something PCR do. Second, hybridization with radioactive probe. This DNA probe and using RNA radioactive probe. This is also not what ELISA do. Third, antigen antibody interaction. So what happened in ELISA? There is antigen and antibody interaction and we can quantify that, and this is how we can detect a pathogen. So the answer is antigen antibody interaction, whereas the fourth option, which is autoradiography to identify mutated genes, no, this is also not the ELISA thing. Next question. Next question asks us, why is gene therapy for ADA deficiency, that is adenosine deaminase enzyme deficiency, involving lymphocytes, not a permanent cure? The question is saying that we use this gene therapy for ADS deficiency, which we have studied in the class, which involves what? Lymphocytes. We genetically altered this lymphocytes and all, but it is not a permanent cure for ADA deficiency. And why it is so? Let's see the options. First option says that the lymphocytes are immortal cells. Immortal cells means that it cannot die, but it is not true. Lymphocytes can die. Second option says lymphocytes have a short lifespan and requires repeated infusion. Required repeated infusions. Yeah, this is correct option, because we know that a lymphocyte is not, it is an immortal cell, it's mortal cell, that it can die, it has a short lifespan, and therefore we require again and again what? We required this again and again infusion, repeated infusion of those genetically modified lymphocytes. So the answer is second option, that lymphocytes have a short lifespan, and it we have to infuse again and again that genetically modified what? Genetically modified lymphocytes. Let's see the third option. Third option says the ADA gene is not correctly inserted into lymphocytes. No, this is not the correct answer for this question. And bone marrow transplantation is more effective. Yeah, bone marrow transplantation is more effective. That is true, but it is not answering this question, that why this lymphocytes is not a permanent cure. Okay. Next question says, how does RNA interference, okay, RNAi, protects plant from nematode infection? We have studied about this RNA interference thing, okay. Interference thing, and we know that it can protect the plants from this nematode infestation. So how it can do this? There have been 4 options. Let's see each option. First option says by producing toxic proteins that kill nematode. Okay. Second, by silencing specific mRNA of the nematode. And third, by enhancing nutrient uptake in plant root. And fourth, by fusing protoplasm with nematode cell. So what is the answer? Answer is answer number second, silencing the specific mRNA of the nematode. That is what RNA interference do. Okay, so answer is second, by silencing the specific mRNA of the nematode. This is what RNA interference do, that it go and bind to the mRNA of that and stop the translation. Next question says, in the production of recombinant insulin using E. coli, why were chain A and B produced separately? Okay, first is, they are asking, means in short, that why this production of recombinant insulin, we have studied in the class that E. coli has separately, we first produce chain A and chain B, and then we create this what? We create this disulfide chain between them. So the first option says that why we do that like that. First option is that E. coli cannot process proinsulin into insulin. Second, the C-peptide interferes with the insulin function. Third, the disulfide bonds form spontaneously when chains are combined. And fourth, that E. coli lacks the machinery to combine the chain. We know that in this production of this recombinant insulin, we use E. coli, and first we differently produce this chain A and chain B, then we create the disulfide bond. Why we do so? First option says that E. coli cannot process proinsulin into insulin. So first option is saying that E. coli don't have this machinery that convert this proinsulin, which has peptide A, B, and C, all those three, into a mature insulin which has only peptide A and B, and that is correct. So answer first is correct, but still we will still all the option. The C peptide interferes with insulin function. That it does. That C peptide is not in the mature insulin, but it is not something why we are producing A and B chain separately. We have to remove it, and that is something that E. coli cannot process. Third question option says that the disulfide bonds form spontaneously when chains are combined. No, we have to create the disulfide bond between them. And fourth options, we can confuse it with first and fourth option. Fourth option says that E. coli lacks the machinery to combine the chain. Okay, E. coli don't have to combine the chain. E. coli has to remove this chain C. So that's why the best appropriate option, which is going to be correct answer, is first only, that E. coli cannot process proinsulin into insulin. Question number 7 says, what is the main advantage of micropropagation in agriculture? That what is the most important, most main advantage, pros of this micropropagation in agriculture? First, it helps in the creation of genetically diverse offspring. No, we know that this micropropagation is used to propagate the same identical copy of the parent plant. Okay. Second option, so first is out of this list because first option is not correct, because it is saying that it is creating genetically diverse offspring. But we know that a micropropagation is used to do what? Second option, that is rapid production of large numbers of genetically identical plants. So the answer is second, but still we will see the other options too. Third says development of pest resistance rate without genetic modification. No. Fourth, reduction in the need for the nutrient media. No, they need a certain nutrient media. So this the right answer is option number second. That is the main advantage of micropropagation in agriculture, is that it can produce large number of the genetically identical plants. Sorry. Then save and next question. Next question says that which of the following is a reason why insulin isolated from pigs or cattle may cause allergic reaction in some patients? So we know that before this recombinant insulin thing, when Eli Lilly company came, it provided the idea that we can extract chain A and B from different this, and then we can join them and all those things. Before that, we used to take insulin from the slaughter pigs or cattle, but they used to cause some allergic reaction in some patients. So why this allergic reactions used to come in some patient and triggers in some patients? So option first said the insulin is not properly processed into mature form. Okay, no, it's not the right answer. So second is the insulin contains C peptide. So we all know that the insulin does not contain C peptide. The mature insulin contain only A and B peptide. Third option says it is a foreign protein, and that's the correct answer, that some patient treat this insulin from pigs or cattle as a foreign protein, and that's how its immune system can get trigger, because it is a foreign protein. Fourth, it cannot form disulfide bond. No, it's not true. So answer is option number third. It is a foreign protein. Now, what is the primary reason for using meristem culture in plant biotechnology? Why we use this meristem culture? Why we use this meristem part of the plant to culture new plants? What is the reason of that? First, to create genetically modified organism. No, we are not genetically modify organism using this meristem culture. to produce virus free plants. Yes, we are using this meristem culture. Why? Because we want to produce the virus free plant, because in this area this cells are always dividing, and because cells are always dividing, there is no chance for the virus to grow in here. Okay, so answer number 2. Let's see the other options. To enhance mineral usage efficiency, no. To develop a somatic hybrid, no. So the answer is to produce virus free plants. Then next question says that which gene is responsible for encoding the beaten toxin in genetically modified cotton? That which gene is responsible for coding what? Bt toxin in genetically modified cotton. So the options are CRY1AC, RNAi, that is RNA interference, agrobacterium, and meloidignine. So these three are not the correct option. Correct option is CRY1AC gene. This is the gene which is going to produce this and code for this Bt toxin. And this is what we insert in that insert in this transgenic plant and all. RNA interference, we know that is used to, for this, we can stop the mRNA translation of nematode, and the nematode is meloidignine. And this agrobacterium is used as a vector for plants. So the answer is CRY1AC. Next question is, which of the following is not a core technique that enable modern biotechnology? So we have studied this in the very first class when we have started this all biotechnology concept chapters. So which is the core technique? Genetic engineering and bioprocess engineering are the two core techniques which is which has enabled this modern biotechnology, and use of this restriction enzyme is also one of the very important techniques in this modern biotechnology. So the what is not here is not a core technique, is the hybridization procedure. So the answer is answer C. Then, how are bacterial cells made competent to take up the recombinant DNA? How we make a cell competent so that it can take what? Recombinant DNA. Options are heat shock at 42 degrees followed by cooling. Second, treating with calcium ions. Third, using microinjection. Or fourth, coating DNA on gold particles. So first of all, answer here is the chemical method which is used for to make the recombinant, to make that host cell competent, we know is using the calcium ions, this calcium chloride treatment. This, don't confuse yourself with this heat shock at 42 degrees Celsius followed by cooling, because this is something we do after treating it with calcium ions. Okay, so here the answer will be, this is also one of the step that is used to, like, first we treat it with calcium, then we heat shock it so that the transient pores in the cell membrane comes. But here we will answer this as treating with calcium ions only, because this is the first step. Okay, after this heat shock step will be followed. No, whereas this using micro injection and coating DNA on cold particles we do in ballistic, all those, in by this we can transfer the recombinant DNA. It is not making the host cell competent. So here the most appropriate answer will be treating with calcium ions. If it wasn't treating with calcium ions, or some other technique has given for this injection of this recombinant DNA into host, then we can go it for first. But when it is giving treating with calcium ions, so we will answer this, because this heat shock at 42 degrees Celsius followed by cooling, this happens after this treating with calcium ions. Save and next. So next question asks us, what is the purpose of origin of replication in cloning vector? We know that one of the important factors we see before selecting this cloning vector is origin of replication. It is this is the point where the initiation of the DNA replication starts. So here let's see the options. First is to identify the transformed cell. No, this is something we for identifying this transformed cell, we use selective markers. So first option, it's not like it something that we can relate to origin of replication. Second, to control the copy number of linked DNA. This is something we have emphasized also and studied during this part also. So, origin of replication can control the copy number of linked DNA. This origin of replication can decide how much copy number of this linked DNA can be formed. So answer is second, but we will see the other options also. To provide antibiotic resistance, no, origin of replication doesn't provide any antibiotic resistance. That there has been other genes for that, and that used as a selective markers. Last, to cut DNA at specific site, no, because this is what means who is responsible for this cutting DNA at specific site. They are restriction enzymes. Question number 14. Which features helps differentiate recombinant colonies using a chromogenic substrate? So in last question also I have told you about selective marker which helps to differentiate these recombinant colonies which has been transformed or not. But which of the selective markers which can be used for identifying this chromogenic substrate, which uses this chromogenic substrate to identify this recombinant colonies? Chromogenic substrate means substrate which is going to change into some color in the presence of some enzyme. Is it antibiotic resistance? No, because antibiotic resistance is going to give the anti-resistance against that antibiotic. No. Presence of sticky end? No, this is something we get after this cutting with DNA, this restriction enzyme and all those things. High copy number origin, this is also not something related to chromogenic substrate. Answer is insertional inactivation of beta galactosidase, because we know that beta galactosidase is the enzyme, is code for an enzyme which can convert this substrate into the blue color colony substrate. So if there has been some gene inserted in between that beta galactosidase gene, it is going to inactivate this gene and there will be no transformation of any color, means no color will be formed. So we can say okay, so there has been transformation in that colony, transformation in that cell. That's why we call it as insertional inactivation of beta galactosidase. But if there has been if there has been no gene inserted in the gene of beta galactosidase, what will happen? It is going to act proper. It is going to convert the substrate into a specific blue color. So we can say that okay, no, there has been no colonies transformed there. So this is how we can differentiate recombinant colonies using a chromogenic substrate. Next question says, which enzyme is responsible for cutting DNA at specific recognition sequence? So they are asking us the name of the enzyme which is responsible for cutting DNA at a specific sequence. So which enzyme is responsible for that? Either it's DNA ligase. No, we know that the DNA ligase is used to ligate to DNA. Restriction endonuclease is the enzyme which is responsible for cutting DNA at specific recognition sequence. Exonucleases, it also cut the DNA sequence, but it cuts at the extreme end, means outside, not at a specific position. Extreme end of the DNA they are going to cut. DNA polymerase is also not responsible for the DNA cutting of the DNA. It is used during replication and all those things. So the answer is restriction endonucleases. Let's see the next question. This question comes from gel electrophoresis. This says, in gel electrophoresis, why do DNA fragments move towards anode? They are asking why, when we run this gel electrophoresis, why this DNA fragments move toward anode. Anode means that positive electrode. Why they are moving toward this positive electrode? Why? First, is the DNA positively charged? No. Agarose gel has a negative charge? No, agarose gel has no charge. But we know that because of that phosphate group, DNA is negatively charged, and that's why it is moving towards the anode. Answer is C. Ethidium bromide imparts a charge. Ethidium bromide has nothing to do with charge. It is for the staining of the DNA. So the answer is third, that DNA is the negatively charged. That's why it is moving towards the positive electrode, that is anode. Now, next question says, which enzyme is referred to as molecular scissors in genetic engineering? It is same as the question before this question, I think 15 or something. But here we are saying that we have given that enzyme a name, which is a molecular scissors. Scissors, we can understand why we use scissors. Scissors we use to cut something. And here molecular scissors has been used because it used to cut DNA at a specific position. So the answer is restriction enzyme only, whereas we know that a DNA ligase is used to ligate two DNA fragments. Polymerase and this DNA polymerase and RNA polymerase are used in their own respective replication process. So the answer is restriction enzyme. Now, question number 18 says, what is the primary function of plasmid vector in recombinant DNA technology? Here again, pay attention to the line that the primary function of plasmid vector. They are asking the primary function of plasmid vector. They are not asking us the function of plasmid or plasmid alone. They are asking the primary function of this plasmid vector as whole. So when plasmid acts as a vector, what is its primary function? Is it acting as molecular scissors? We know that no, restriction endonuclease are something which is acting as this molecular scissor. Then to replicate independently in the host, we know that this plasmid has the ability to replicate independently in the host. They are extra chromosomal, they are not a part of the DNA chromosome. But here again I am saying we have to emphasize on the question that the question says primary function of a plasmid vector. So let's see the other option also. So, the primary function of a plasmid vector is to transfer the foreign DNA into host organism. So, the answer is option number C. Fourth, to sterilize the bioprocess environment. No, it is not used to sterilize the bioprocess environment. First and fourth option is definitely a no. But second and third, and you might get confused, but don't get confused because they are asking the primary function and the function of plasmid vector. When plasmid act as a vector versus primary function is, so its primary function is to first they transfer the alien DNA into the host organism. Save and next. Then next question says that why is traditional hybridization considered less efficient than genetic engineering? Kind of, they are asking the drawback of traditional hybridization and why it is less efficient than genetic engineering. So first, it cannot create recombinant DNA. No, it can create recombinant DNA. It often introduce undesirable genes. So the reason is that they are not genetically engineered, they are not very much controlled, so they can often produce the undesirable genes. That's why it is less efficient. It requires bioprocess engineering, and it cannot use plasmid as vector. No, these are not the correct options. The correct options is that it often introduces the undesirable gene, and that is why this traditional hybridization is less efficient than genetic engineering. Let's see the last question. So last question says us, what is the role of origin of replication in genetic engineering? So there was a question which was asking about this copy number, which is related to the origin of replication, but here they are asking the role of origin of replication in genetic engineering. So first option is only the answer, because the role of origin of replication in genetic engineering is to initiate the DNA replication. To join DNA fragment is the role of DNA ligase. To cut DNA at specific site is the function of restriction endonucleases, and to transfer DNA into host is the role of vector. So the answer is origin of replication in genetic engineering is used to initiate the DNA replication, and they can also control the copy number. So let's save. Okay, we will submit our test, and yes, this time, yes, we have to submit. We can see that all of our answers are correct and we have submitted in time also. So now we will start with chapter 10, Biotechnology and its application, some practice questions. So let's start the practice questions. This is a disclaimer. So the learning objective is going to be like this, that we will understand and explore the fundamental concept of the chapter 10, Biotechnology and its application, by working on some practice questions. So index will be that we will be doing C practice question from 1 to 22 from chapter 10, Biotechnology and its application. So first question says us, which bacterium produce Bt toxin? Options are Escherichia coli, Bacillus subtilis, Bacillus thuringiensis, or Pseudomonas butida. So the answer for this question is that Bacillus thuringiensis. We know that this Bacillus thuringiensis is the bacteria which can naturally produce the crystal proteins, okay, which is a toxin to insects. These proteins are called Bt toxins, and these toxins have been used to create pest resistance GM crops. They are in the inactive form when they are present in the bacteria, but they can get activated when they go inside the alkaline nature of insect's gut. So examples include Bt cotton, corn, rice, tomato, potato, etc. This is a pictorial representation of Bacillus thuringiensis, where we can see that there has been this Bt toxin crystal, and this alkaline pH, when they go inside this insect, this alkaline pH has the ability to dissolve this crystal, and then this Bt toxin can get activated. Next question says, the process of growing plant cells tissue in artificial medium under sterile condition is called. So when we can, in sterile condition, when there is no contamination, nothing, we can grow the plant cells and tissue in artificial medium. So which that process is called? Options are tissue grafting, tissue culture, tissue engineering, or tissue transplantation. Answer B, tissue culture. It involves what? In growing explant. Explant means any part of the plant. So it involves explant growing in nutrient medium under sterile condition. That nutrient mediums consist of what? They have some special growth medium with nutrients, vitamins, hormone, and all. This is based on the principle of totipotency. Totipotency we have talked in today's class, that the potential of a cell to mature and to grow into any kind of cell, and then became a whole plant. So this is the principle of totipotency, then used for micropropagation and producing disease free plants. So this is the pictorial representation of a plant tissue callus, where we take a plant, we have take some part of that plant called explant, then we can grow that explant in some nutrient media, then that will form callus, embryo, and then finally it will become plantlet and then can grow into a plants. So now what is RNAi? RNAi is either what? RNA isolation, RNA interference, RNA integration, or RNA inheritance. So the answer is that RNAi is RNA interference. This is what? It is a natural cellular defense mechanism present in eukaryotes. It involves what? So it involves silencing of specific mRNA using the complementary double stranded RNA. Okay, and it does what? It will go and bind to the complementary DNA, and then complementary mRNA, and prevents the translation of target mRNA. It is used to develop the pest resistance plant, specifically nematodes. You guys can see here is the action mechanism of RNA interference. So there is like a lot of thing here, but what you guys can understand is that this double stranded RNA, that could be anything: siRNA, that is small interference RNA, or miRNA, that is the microRNA. Okay, this dsRNA, that is siRNA and microRNA, they can work with this, along with this with the help of this enzyme dicer. Okay, it can convert into this with the help of the dicer. What happens? It can convert into this, and then it forms this risk, that is RNA induced silencing complex. Okay, and this complex they form how? So this complex they form by this siRNA or mRNA, microRNA. It combines with the organoid protein. So this protein is going to make this siRNA. Then what it is going to do, it is going to um, unwind it and make this complex active. And then what happened, this guide strand it has been shown here, it will go and binds to the complementary part of the mRNA, and this is how it is going to stop the translation. So this is how it is going to work. I don't think it is in this much detail in NCERT, but still you guys can understand, okay, that this how we can, with the help of this action of this enzyme dimer, we can derive this siRNA and microRNA, and then this along with the argonauts protein going to form this RISC complex. This RISC complex has this guide RNA which is going to go and bind to this mRNA and stop the translation. Now, next question says that the first clinical gene therapy was given for which disorder? So, the first gene therapy we have studied that has given to a 4 years old girl is against which disorder? Is it cystic fibrosis, sickle cell anemia, ADA deficiency, or thalassemia? So, the answer is ADA deficiency, that is adenosine deaminase deficiency. It is a first gene therapy given to a 4-year-old girl in 1990. With why it is crucial, why this enzyme is crucial? This enzyme is crucial for immune system functioning, and this is the whole what it is a whole diagrammatic representation of how we have taken this T cell lymphocyte T cells, okay, from the patient, then we have cultured that T cells in the patient, then at the very parallel end what we are doing, we will taking the bacterium which is carrying the DNA vector with cloned normal ADA gene. Then with the help of this vector, which is a retrovirus, which is a disabled retrovirus, we will do what? We will going to infect that T cells which we have culture of that patient. And then what will happen? This T cell will be, this T lymphocytes will do what? So this will genetically modified with the help of this retrovirus with this DNA which is cloned with normal ADA gene, and then we will reinfuse that ADA gene containing T cells, genetically modified cells, back into the SCID patients. SCID patients are the patients which has this lack of this ADA enzyme, and like that we can cure this, not permanently cure this, but that's how we can treat the ADA deficiency using this gene therapy. Then it's involved in reducing functional ADA gene into lymphocytes. As I told, it required the periodic infusion of engineered cells. Why? Because we know that the T cell, that lymphocyte cells, they have shorter lifespan, and they are going to die. Then next question says, what is the main advantage of producing insulin through genetic engineering? So what is the most important advantage of this producing the insulin from genetic engineering? So either is it cheaper production, or no immune response, or better effectiveness, or easier administration? So the answer is answer B, that no immune response. So but the main advantage of producing this insulin through genetic engineering is that they are not causing any allergic reactions, because this genetically engineered human insulin is identical to the natural insulin, and human body is not going to treat it as foreign substance, and therefore it is not going to trigger any immune response. It can be produced in large quantities also using the bacteria, and that's why this is important advantage. It is in part advantage of producing insulin through genetic engineering. Next question says, what is biopiracy? Either it is what is a biopiracy? Is it stealing biological samples? No. Unauthorized use of bioresources? Okay, so yes. So biopiracy is the unauthorized use of bioresources. Illegal trade of organism, no, it is not biopiracy. And copying biological process is also not biopiracy. It is the unauthorized use of the bioresources. So, this unauthorized exploitation of the biological resources is what we call biopiracy. It is often done by whom? It is done by multinational companies without the proper compensation. Biopiracy affects the developing countries, which is rich in biodiversity, and it includes patenting traditional knowledge without authorization. So, here's the picture that they're stealing our bioresources. Then next question says, which technique is used for early detection of pathogens? So this is something we have also discussed in the mock test also, but here is that we have to name the techniques which we can use to detect pathogens earlier. Either is ELISA, that is enzyme linked immunosorbent assay, either is PCR, that is polymerase chain reaction, either it is both, that both A and B, or neither A or nor B. Let's see the answer. Answer is answer C, both A and B. Why? PCR do what? PCR amplifies pathogen nucleic acid, that is DNA and RNA. Even if we have a very small amount of DNA or RNA of that pathogen present in our blood or in our body, PCR going to identify it because it can amplify that pathogen DNA or RNA. ELISA did what? It helps in detecting antigen or antibody. If we have antigen inside our body, it can also detect that, or even if we have made antibodies against that antigen, it can also detect that. So this is the principle of PCR. So they have shown what are the different steps of the PCR that we have discussed in very detail in the classes. First, denaturation step, in which the double stranded DNA is going to denature. Then primer will come and going to anneal. Okay, the second step, annealing. Then the DNA polymerase, specifically Taq polymerase, because it has the ability to work even in the higher temperature, it is going to amplify this whole region, and this is how we can make the very high number of DNA. It has been shown that in like after 30 cycles we can make approximately it became 1 billion times. So this is the principle of PCR. Then why this both techniques is used? Both allow early diagnosis before symptoms appear. Even if there is no symptoms in the body, they can appear, they can show the diagnosis ki yes, these people are infected with those disease. Widely used for disease diagnosis. This is against the principle of PCR. Next question says that the process of combining protoplast from different plant varieties is called, means we are taking the protoplast from the different plant variety and fuse it. What that process is called? Either is it called hybridization, either is called crossbreeding, or somatic hybridization, or genetic modification. So it is called somatic hybridization. So what happens in somatic hybridization? It involves the fusion of protoplasm from the different plants. It creates somatic hybrids, means hybrid of two somatic cells. So here you guys can understand it more clearly. What they did, they have isolated these cells from the two different plants. They do what? They remove their cell walls. Okay, and now what remains? The protoplasm. Protoplasm means cytoplasm plus organelles or the nucleus. Now they will take the protoplasm of both the cell, and fusion of this protoplasm will occur. After the fusion of those protoplasm, what will be come out? The outcome will be there will be somatic cells hybrid. This will stimulate division and differentiation of those hybrid, and this now hybrid leaves can grow. This is what we call protoplasm fusion. So example is pomato, which is the protoplasm fusion of what? Potato and tomato. Different from sexual hybridization. This is not something, it is very different from sexual hybridization. It is protoplasm hybridization. That's why we call it somatic hybridization. Then next question is like, what is the role of Genetic Engineering Approval Committee? So what is the role of GAAC in India? Options are it does patent approval, Is it for research funding? Is it for safety assessment of GMO? Or it is for technology transfer, safety assessment of genetically modified organisms or technology transfer? So the answer is option number C. It is for safety assessment of GMOs. So the role of the genetic engineering approval committee is that it is a government regulatory body. It evaluates the GM, that is genetically modified research validity, and the safety assessment of this GMOs so that it can go to public services, make decisions about the GMO introduction into the environment, and that's why this committee has been made. Next question says that which is not an application of transgenic animal, means among them which is not an application of transgenic animals. Is it that we cannot use the transgenic animal as disease models or biological product or vaccine testing or food production? So the answer is option D, food production, because we know that we can use the transgenic animals for, as a disease model, for biological product, or as a vaccine safety model also. So the transgenic animals mainly used for research, they are used to study diseases and treatments, then they are aimed to produce therapeutic proteins, and these transgenic animals are not primarily developed for food. So the answer is option D, food production. Then the question says the first transgenic cow was named. So we have studied about that in 1997. We have made the first scientists have made the first transgenic cow, and what was the name of that cow? Is it Dolly? Is it Rosie? Is it Tracy? Or is it Molly? So the answer is it is Rosie. It is she's the first transgenic cow created in 1997. What was the benefit of this cow? That it can produce the human protein, that is alpha lactalbumin enriched milk. It can produce 2.4 gram of this protein per liter of her milk. Better balance for human babies than the normal cow milk. The next question says that which component is removed during insulin maturation. So this is something we are studying for so long that, but see the option A chain, B chain, C peptide, or disulfide bonds. We know that answer will be C peptide. Why? Because we know that insulin synthesized has prohormone C peptide present in proinsulin, and to make it a mature insulin we have to remove the A and B peptide. We have to remove the C peptide and keep this A and B, which is linked together with disulfide chain. Now this C peptide is removed during maturation. C peptide not present in the functional insulin. So the answer was C peptide. Next question asks us, what is micropropagation? Options are small scale farming, tissue culture based plant multiplication, genetic modification, or selective breeding. So the answer is that micropropagation is a tissue culture which is based on plant multiplication. So what is micropropagation? It is the mass production of plants through tissue culture. It produces what? It produces genetically identical plants that are soma clones. So this is the figure which shows what? So what they did, they have taken a source plant, they have taken this, so some part, small part, that is also called explant. There's a bulb corn of source plant, then we did the primary treatment, then there will be reproduction and multiplication. First shooting will happen, then rooting and all those things, then there is primary hardening, then there's secondary hardening, and then again it will become the source plant, identical to the source plant. This is what we call micropropagation. It is a rapid multiplication method. It used commercially for many food plants, example banana. Next question says ask us which part of the plant is virus free and used for tissue culture. Means which part of the plant is what? Virus free, means there is no virus in there and we can use it as tissue culture. Is it leaves? Is it roots? It is meristem or stem? So we have studied this type of question also in mock test, that it is meristem. It is apical and axillary meristem which are virus free. They produce virus free, so they can use to produce virus free plants. They can divide rapidly, and this rapid cell division in this portion did what? It doesn't allow virus to infect. So this is the apical meristem and this is the root apical meristem. It is, yeah, this is apical meristem, this is the shoot apical meristem, and this is the root apical meristem. Next question says golden rice is enriched with. We know that golden rice is a genetically modified crops. It is made to, and it is made to what? It is made with, to make it enriched with some nutrient. That nutrient is what? Is it protein or vitamin A or iron or zinc? Answer is vitamin A. Golden rice is genetically modified rice variety. It is created to enhance what? Nutritive value. It contains beta carotene, which is a vitamin A precursor. It is going to convert into vitamin A when it comes to our body. Addresses vitamin A deficiency, and this is how it looks. Then, next question says that which of the following is not a component required for tissue culture media? We know that for this tissue culture medium, we need a medium. That media has many things, but among this, what is not needed, which is not a component which is required in that media? Is it sucrose? Is it inorganic salt? Is it growth hormones, or it is antibiotics? Answer is antibiotics, because we know that the important components required for tissue culture medium, first it is carbon source, sucrose, it is the energy source, then it has some inorganic salt and vitamins which are required, then there has been some growth regulators like auxin or cytokinins which is required, which is help in the growth of the plant. Antibiotics are not a standard requirement for tissue culture medium. It may be required, but for some plants or some anything, but it is not a standard requirement for tissue culture media. Next question says that what are genetically modified organism? Is it naturally evolved organism? No. Then is it organism with altered gene through manipulation? Yes. Is it hybrid organism? It is mutated organism. No. So this is the organism which is altered, whose gene has been altered through manipulation. So answer is obviously option B. So GMOs, that is genetically modified organisms, have deliberately did what? Modified their genetic material. It can include plant, bacteria, fungi, animals, anything. Modification is done through what? It's done from the genetic engineering techniques. So this is a transgenic mouse which has been shown in the figure. What is totipotency? Ability to generate new organs, ability to generate a whole plant from any explant and cell, ability to produce flower, or ability to produce fruits. We have studied a lot of time in today's class, in even mock test, that it is the ability to generate a whole plant from any cell or explant. Totipotency is what? Is a capacity of even a single cell to generate a complete plant. This is achieved under sterile condition in this nutrient media. Sterile condition means contamination-free, micro-free environment in a special nutrient media. This process requires specific nutrients, hormones, and growth conditions. Thank you.